So a monohybrid cross is a genetic cross where the parents are different from one another in one way. And in this cross, we're going to look at a tongue roller, the characteristic, the gene for tongue rolling. And to do a monohybrid cross, we need to use letters to represent the alleles. Remember, alleles are different forms of a gene. So the dominant allele is tongue rolling. So I'm going to use a capital R to represent the tongue roller allele. And the non-tongue roller is the recessive characteristic. So I'm going to use a small r to represent the non-roller. So all monohybrid crosses start with a parental generation. And this is where you cross two individuals who are homozygous for the different characteristics. So one parent is a homozygous tongue roller and the other parent is a homozygous non-roller. So put a wee p at the side to represent the parents, and the parental genotype is big R, and we're crossing that with a parent who's a non-roller, so they're homozygous small r, small r. So the first thing we have to consider is what the gametes for the parents are going to be, because remember, all cells in the body are diploid, but when we produce the gametes, they're haploid, which means they have half the genetic information. So all of the gametes that our tongue rolling parent produces are going to be containing the capital R, the tongue rolling allele. All the gametes that our non-rolling parent are going to produce is going to be small r. So when that gamete fertilises that gamete, we are going to get our first generation, and our first generation are all going to be big R, small r. That means that they are all heterozygous because the two alleles are different. Right, so they're all heterozygous, and they are all going to be able to roll their tongues because the tongue rolling allele is dominant, and one copy of the dominant one means that's what's going to appear in the phenotype. They are carriers for the non-rolling allele, but that will only show up in the phenotype if you are homozygous for it, i.e. if you have two small letters. So the next stage of our monohybrid cross is to do what we call an F1 cross. And all that means is that you're taking two members of the first generation and crossing them. Okay, so you are taking a big R, small r, and you're crossing that with a big R, small r. Same as previously, we need to work out what the gametes are going to be. So in this case, this parent has got two different gametes. So half of the gametes that they produce are going to have a big R in them, and half of the gametes that they produce are going to have a small r in them. And obviously the exact same for this parent. Half the gametes are going to be big R and half the gametes are going to be small. So when those gametes get together, there's a variety of different combinations that we can get. So to work out all of those possible combinations that are going to be in the second generation, we draw what's called a Punnett square. And in the Punnett square, we just take the gametes from this parent and put them down one side. So that would be a big R and a small r. And then for the same from this side, so a big R and a small r. And what we're doing now is working out, well, if this gamete meets this gamete, then the genotype that would result would be an individual with big R, big R. They are homozygous for tongue rolling, and they would obviously be able to roll their tongue. If this gamete meets this gamete, we've got a big R and a small r. So that's a heterozygous individual who can roll their tongue. That's exactly the same as the parent from the first generation. Same here, big R and small r. And then if these two meet, then we get a homozygous individual who cannot roll their tongue because they've got two copies of the recessive allele. So we do something called now an F2 phenotypic ratio. Right? And all that means is we're looking at the ratio of phenotypes in the second generation. So this individual, and this individual are all going to be able to roll their tongue. So that's 3 tongue rollers. And then we've got this individual here, that is the non-roller, so that's 1 non-roller. And that's a monohybrid cross. And every single question that you get on monohybrid crosses relies on you being able to do this. And it doesn't matter what letters or what characteristic we look at. So for example, we could do earlobes attached and non-earlobes attached. And then, so you could have big E, small e, but that pattern is going to be exactly the same. Now, we're saying that if we do all of this cross and we get to this point, 75% of our offspring should be able to have the dominant characteristic, and 25%, 3 to 1, 75-25, would have the recessive characteristic. That's not always the case. Sometimes there are predicted offspring, i.e. this proportion here is different, and that's because fertilisation is random, okay? So you might get 60-40 split. If it, for example, if there was 100 plants, you might get 60 that had the dominant characteristic and 40 that had the other, and that's just because fertilisation is a random process. So the other question that comes up quite often for monohybrid crosses is, why are predicted ratios different from actual, right? So the difference between predicted and actual is because fertilisation is a random process. So difference between predicted ratios and actual ratios is because fertilisation is a random process. Okay, and that is a monohybrid cross from start to finish. Parents, gametes, first generation, F1 cross, second generation, F2 phenotypic ratio, and then knowing the difference between predicted and actual ratios.